I want you to picture the most secure facility on Earth. Think the Svalbard Global Seed Vault, buried in the Arctic permafrost, or maybe the secret archives of the Vatican. It's temperature controlled, hermetically sealed, and just incredibly static. And in the very back, inside a titanium box, lies the master instruction manual for a human being. That is DNA. It is the reference copy. And the number one rule of this facility, the reference copy never ever leaves the vault.
Never. Because if you scratch that reference copy, you don't just ruin one page. You ruin the instructions for every single subsequent generation. You corrupt the entire lineage. The number one priority for DNA is preservation. It has to be like geologic in its stability.
Right, but a civilization or, a cell can't function on a book that just sits in the dark. You need to build things. You need bridges, enzymes, pumps, neurotransmitters. You need to get the information out of the vault and to the actual job site. So you need a scribe, someone to go into the vault, open the book, and furiously scribble down a copy of page 42. And that copy, that's RNA.
And that distinction right there that sets up the entire drama of cellular biology that we're exploring today. DNA is all about storage. RNA is about execution. It's messy, it's volatile, it's heavily edited, and as we're going to find out, it is absolutely destined to die.
So today we are opening up the hood on that whole process. We are doing a deep dive into RNA metabolism. We pulled two heavyweights for the synthesis. We're looking at Lehninger principles of biochemistry, specifically that chapter on RNA metabolism to get the molecular nuts and bolts.
The mechanics.
The mechanics. And we're pairing that with Harper's Illustrated Biochemistry, the chapter on RNA synthesis, processing, and modification, to really understand the clinical reality, what happens when this machinery jams up.
Right. We're using Lehninger to visualize the chemistry, the sheer beautiful mechanics of how you stitch these molecules together. And we're using Harper's to ground it in medicine. Because when RNA metabolism fails, you don't just get a typo. You get diseases like spinal muscular atrophy or lupus.
And a quick reality check before we start. We are synthesizing a massive amount of dense biochemistry here. So think of this conversation as like the aerial map. We're going to show you the landscape, the mountains, the rivers, the trade routes. But you still have to hike the trail yourself. This doesn't replace the textbook, it contextualizes it. We want you to go back to Lehninger and Harper's and look at those diagrams with fresh eyes.
And that context is absolutely crucial. I think the biggest mistake students make is thinking that RNA synthesis transcription is just copying. It's not. It is curation. The cell edits the information. It censors it. It adds things. It modifies it. The RNA molecule that leaves the nucleus looks very, very different from the DNA gene it came from.
It's the difference between the raw footage and the final cut of a movie. So let's meet the director, or I guess the writer. We need an enzyme that can break into the vault. In our deep dive on DNA replication, we talked about DNA polymerase. Here we have the DNA-dependent RNA polymerase. Let's just call it RNAP.
RNAP is a fascinating machine. If you remember our discussion on DNA replication, DNA polymerase was a bit neurotic. It required a primer. It couldn't start from scratch. It needed a little landing pad of existing nucleotides to bond to. It was kind of needy.
Right, it couldn't initiate. It could only elongate what was already there.
RNA polymerase is a cowboy. It initiates de novo. It grabs the first nucleotide, grabs the second, and just forces them together. No primer, no looking back. It just starts writing.
Okay, but does that confidence come with a cost? I remember DNA polymerase having that almost obsessive proofreading capability, the 3-foot to 5-foot exonucleus activity that was like a backspace key.
It absolutely comes with a cost. DNA polymerase makes a mistake maybe once every 10 million base pairs.
Yeah.
It's incredibly accurate. RNA polymerase, it messes up about once every 10,000 to 100,000 base pairs. It's significantly sloppier.
Wow, that sounds dangerous. If I'm building a bridge, I don't want a sloppy blueprint.
It's a calculated risk. If you mutate DNA, that error is permanent. It's burned into the hard drive. It gets passed to every single daughter cell. If you screw up one RNA molecule, well, you make one bad protein from it, maybe a few bad proteins, and then the RNA gets degraded a few hours later. It's a temporary glitch, not a systemic failure. The cell is prioritizing speed and responsiveness over absolute geologic perfection here.
Let's look at the engine itself then. How does it actually forge that bond? I assume, since this is biochemistry, we are dealing with our old friend magnesium.
It's always magnesium. Lehninger is very specific about this mechanism, and it's beautiful in its simplicity. Deep in the active site of the enzyme, you have these conserved aspartate residues. Aspartate is an amino acid with a negative charge. And these residues, they hold on to 2 magnesium ions, 2 Mg2+ ions.
And their job is to mess with the electron balance, right? To make the chemistry happen.
They act as chemical leverage. So you have the growing RNA strand, and at the very end, there is an oxygen molecule on the 3' carbon, a hydroxyl group. One magnesium ion coordinates with that oxygen and makes it what we call nucleophilic.
Which means it makes it chemically aggressive. It wants to attack something.
Exactly. It wants to attack the phosphate group of the incoming nucleotide. The magnesium makes that attack possible. Then the second magnesium ion comes in and helps stabilize the whole reaction and kicks off the pyrophosphate group, which is just the waste product. So without these two magnesium ions held perfectly in place by those aspartates, transcription just stops, dead in its tracks.
So it's a push-pull mechanism, and it's all mediated by metal ions. And this is all happening inside what we call the transcription bubble.
The bubble is absolutely key. The enzyme creates a localized unwinding of the DNA duplex. It's about 17 base pairs long.
I've seen the diagrams in Leninger. It looks like a zipper just opening up a little bit.
That's a good visualization, but add this one detail. Inside that bubble, for a very brief moment, the new RNA strand is physically stuck to the DNA template strand. It creates a little DNA-RNA hybrid helix, but it's super short, only about 8 base pairs. As the enzyme moves forward, a structure inside the polymerase acts like a wedge, or a rudder, to peel the RNA off so the DNA can zip back up behind it.
It's a factory line. Material comes in, gets stamped, and then gets peeled off the mold. Now, the machinery doing this differs depending on whether you're a bacterium or, a human. Let's start with the simpler model. E. coli.
Right. So bacteria use what we call a holoenzyme. You have the core enzyme, which is a bunch of subunits, alpha-2, beta, beta-prime, omega, which does the heavy lifting of actually building the RNA. But here's the problem. The core enzyme is blind. It has no idea where to start on the genome. It would just bind randomly to the DNA and make absolute nonsense. It needs a guide. And the guide is the sigma factor.
So the sigma factor is the navigator. It's the GPS.
It's the GPS. It scans the DNA looking for the promoter, the address that says start gene here. Specifically, it looks for consensus sequences at the -10 and -35 regions upstream of the gene. Once the engine is running and the RNA is being made, the sigma factor often just falls off. Its job is done.
But here's the strategic brilliance of bacteria that I want to dig into. They don't just have one sigma factor, they have a whole wardrobe of them.
And this is how they survive such rapid environmental changes. Under normal conditions, they use a factor called sigma 70. Sigma 70 takes the polymerase to all the housekeeping genes, the stuff needed for day-to-day life, metabolism, cell wall maintenance, things like that. But let's say you crank up the temperature. The bacteria is cooking.
A heat shock.
A heat shock response. The cell suddenly stops using sigma 70 and swaps in sigma 32. This new navigator, sigma 32, ignores the housekeeping genes. It drives the polymerase exclusively to the heat shock genes that make chaperone proteins to protect the cell's other proteins from burning up. It essentially changes the entire operating system of the cell instantly just by swapping out that one little protein subunit.
That is just ruthless efficiency. It's like switching cartridges in an old Nintendo. Same console, totally different game. Now, humans, eukaryotes, we never do anything that simply. We don't have just one polymerase, we have 3.
We have a division of labor. It's much more specialized. You have RNA, polymerase of first, pol 2, and pol 3.
The mnemonic I always use is RMT. Pol I makes ribosomal RNA, pol 2 makes messenger RNA, and pol 3 makes transfer RNA.
That holds up really well. Pol 1 is the factory builder. It makes the parts for the ribosome. Pol 3 is logistics. It makes the tRNA trucks that carry amino acids. But pol 2, pol 10 is the celebrity. It's the one that makes the mRNA. It's the one reading the genes that determine your eye color, your insulin levels, your neurotransmitters. It's reading the actual recipes for proteins.
And because pol 10 is so critical, it's a prime target for assassination. Harker's tells a pretty grim story about the death cap mushroom.
Yeah, Amanita phalloids.
Ugh.
It's this beautiful mushroom, looks totally innocuous, but it contains a peptide called alpha-amanitin. This toxin is basically a heat-seeking missile for eukaryotic pol 10s. It binds to a specific part of the enzyme, and it essentially clamps the jaw shut. It presents translocation. The enzyme literally cannot move down the DNA.
So it freezes the transcription bubble in place, the zipper gets stuck.
Exactly. It's an instant paralysis of the information flow. You stop making new mRNA. No new instructions means no new proteins. The cell literally runs out of working parts. The liver takes the first and hardest hit because it's trying to filter the toxin. It's a slow, gruesome way to go because you are essentially starving your cells of new instructions.
But, and this is key, it doesn't kill bacteria.
No, because their polymerase is shaped differently. The toxin can't bind. And that fundamental difference is the basis of so much of modern medicine. Antibiotics like rifampicin work on the exact same principle. But in reverse, they jam the bacterial polymerase while leaving our human polymerase completely alone. It's chemical warfare based on structural geometry.
Okay, so let's zoom back into a human cell. POLDUS is chugging along, making an mRNA copy, but this isn't like printing a document where you wait for the last page to slide out of the printer before you staple it. The processing starts while the ink is still wet.
Co-transcriptional processing. This is a huge concept in literature. The RNA polymerase II isn't just a scribe, it's a workbench. It has this long, flexible tail called the CTD, the carboxyl terminal domain.
Linenger describes this tail as unstructured, so it's just sort of flapping around back there.
It's flopping around until it gets phosphorylated. This is the key. As soon as transcription starts, kinases come in and add a bunch of phosphate groups to the amino acids on this tail. And once those phosphate groups are added, the tail becomes a docking station. It's a recruitment platform. It recruits all the enzymes that are going to edit the RNA. They ride on the tail of the polymerase like passengers on a bus, just waiting for the new RNA to emerge so they can jump on it.
So the moment the 5' end, the very beginning of the RNA pops out of the enzyme, the first team jumps off the tail and modifies it. This is the cap.
The 5' cap. It's a bizarre structure. Usually biological chains link head to tail, 5' to 3', but the cap is a guanosine nucleotide that's linked 5' to 5'. It's this weird triphosphate bridge. Essentially, the cell puts a nucleotide on backward.
Why? Is it a disguise?
It's armor. It's absolutely armor.
Yeah.
The cell is full of enzymes called exonucleases that just patrol the cytoplasm, looking for loose RNA ends to chew up. They're the cleanup crew. They recognize a standard 5' end. But this backward methylated cap, they don't know what to do with it. It's chemically invisible to the shredders.
But it's very visible to the ribosome.
It's essential for the ribosome. It's the VIP pass for translation. If an RNA doesn't have a cap, the ribosome will not touch it. This is actually how the influenza virus messes you up so badly.
The cap snatching mechanism.
It's brilliant, and it's terrible. The flu virus creates its own RNA genome inside your nucleus, but it can't make caps. It lacks the enzyme. So to have a viral endonuclease that literally hunts down your newly made host mRNAs, your cellular messages, and it just cuts the caps right off them.
It decapitates your own messages to masquerade as self.
Exactly. It takes that stolen cap, stitches it onto its own viral RNA, and sends it out to the ribosome. Your cell then protects the viral RNA and translates it, thinking it's a friendly message. Meanwhile, your own uncapped mRNAs get found by the exonucleases and destroyed. It's molecular piracy.
That is just ruthless. Okay, so that's the front end, the 5' cap. Now let's talk about the back end, the 3' end. The transcript is finished, the enzyme cuts the RNA loose, but then it adds something that wasn't in the DNA blueprint at all, the poly-A tail.
This is a string of somewhere between 80 and 250 adenosine residues, just A, over and over. And you're exactly right. There is no corresponding string of T's in the DNA. A specialized enzyme, polyadenylate polymerase, just builds this tail onto the end after the transcript is cleaved from the polymerase.
I always tend to think of the poly-A tail as a fuse on a stick of dynamite.
That is the perfect analogy. It is a timer. The moment the mRNA hits the cytoplasm, enzymes start nibbling away at that tail, shortening it 1A at a time. And when the tail gets too short, that's the signal. Time's up. The protective proteins that bind to the tail fall off, the cap is then removed, and the mRNA is targeted for degradation.
So the length of the tail directly determines the lifespan of the message. A longer fuse means more protein production before the whole thing goes boom.
Precisely. It allows the cell to tune exactly how much protein it gets from a single gene activation. If you need a protein for a long time, like beta-globin, you give it a nice long tail. If you need a quick burst of a protein for just a few minutes, you give it a short one.
Okay, we've armored the front, we've timed the back. Now we have to deal with the absolute mess in the middle. This is the part that always just blows my mind about eukaryotic biology. We transcribe huge amounts of DNA that we have zero intention of ever keeping.
The introns, the intervening sequences. If you look at the gene for dystrophin, the one involved in muscular dystrophy, it spans 2.4 million base pairs of DNA. It is an absolute leviathan. But the final functional mRNA, the instructions that actually build the protein, it's just 14,000 bases.
Wait, what is that? Less than 1%?
We throw away 99.5% of the transcript. We transcribe it, which is energetically expensive, and then we just cut it out and destroy it.
From an engineering standpoint, that seems incredibly wasteful. Why not just have the gene be 14,000 bases long and skip the middleman? Why transcribe all the junk?
Because of what we call combinatorial power. By having the gene broken up into these little islands of information, the exons, separated by these vast oceans of introns, you can mix and match. In one cell, you can stitch exon 1 to exon 2 to exon 3. But in another cell, you can skip exon 2 entirely and stitch 1 directly to 3.
Alternative splicing.
And suddenly, one gene isn't one protein. One gene is a kit of parts that can build 20 different proteins depending on the tissue or the developmental stage or the time of day. It helps explain how humans can be so complex, how we can build brains and immune systems, despite having only about 20,000 genes, which is roughly the same number as a microscopic worm.
So the waste actually buys us complexity. It buys us a library of options. But the physical act of cutting this splicing has to be incredibly dangerous. If you miss the cut by a single letter, you shift the entire reading frame and you ruin the protein. How does the cell execute this cut with such precision?
Welcome to the splicing. It's arguably the most complex single machine in the cell. It's made of these subunits called snRNPs, which everyone pronounces SNRPs.
Small nuclear ribonucleoproteins. Quite a mouthful.
It is, but they contain these small RNA molecules, U1, U2, U4, U5, U6, that are the real key. They recognize the boundaries of the intron. They literally base pair with the start and the end of the section that needs to be cut. And then they perform this incredible piece of molecular origami. They bend the intron into a loop.
The lariat.
Yes, they take a specific adenosine residue that's sitting inside the intron, and they force it to attack the 5' start of the intron. It forms a literal, not a chemical lasso. This frees the first exon. That free exon then swings around and attacks the start of the second exon, joining them together. And the intron is released as this lariat loop, and it's sent off to be degraded and recycled.
Now, Leninger points out something here that fundamentally changed our entire understanding of evolution. The protein parts of the spliceosome, they aren't the blades of the scissors, are they?
No, they're just the scaffolding. They hold everything in the right place. The chemistry, the actual cutting and the stitching, is catalyzed by the RNA components, specifically U6 RNA. The spliceosome is a ribosome. It's an RNA enzyme.
Which just screams RNA world.
It screams it. It's powerful evidence for the hypothesis that before DNA and proteins took over, life was ruled by RNA. RNA carried the genetic code, A and D RNA, did the chemical work. The spliceosome is like a living fossil from 4 billion years ago, still chugging away in all of our nuclei.
But when this ancient machine slips up, the consequences are very modern diseases. Let's talk about spinal muscular atrophy, SMA. This is the leading genetic cause of infant death, isn't it?
It is, and it's a pure splicing tragedy. To understand SMA, you have to know that all humans have a gene called SMN1. It makes the survival of motor neuron protein. You absolutely need this protein to keep your nerves connected to your muscles.
And SMA patients have a defect in this gene.
They're either missing SMN1 or it's mutated and non-functional. But here's the kicker. They have a backup copy, a gene called SMN2. It's a duplicate gene that's sitting right next door.
So why doesn't the backup save them?
Because of a single so-called silent mutation? In the SMN2 gene, there is one letter change. A cytosine becomes a thymine, a C to a T in exon 7. Now, if you look at the genetic code, the codon with a C and the codon with a T both code for the same amino acid, so it shouldn't matter for the final protein structure.
But it matters to the spliceosome.
It matters everything to the spliceosome. That C was part of a landing pad, an exonic splicing enhancer. It was a little signal that told the spliceosome, hey, keep this exon. This part is important. When it changed to a T, that signal vanished. The spliceosome stopped seeing exon 7 as a true exon. Most of the time, it just skips it.
So the backup gene makes a protein that's missing a crucial piece. It's unstable and just falls apart.
Exactly. So you have a patient with a broken main gene and a backup gene that is being systematically spliced incorrectly. The motor neurons die. But we have a fix now, a drug called Spinraza.
Nusinersen.
It is a triumph of biochemistry. It's a small piece of modified RNA and antisense oligonucleotide. You inject it into the spinal fluid, and it goes into the neurons, and it binds to that exact spot on the SMN2 transcript. And it mimics the missing signal.
Yeah.
It essentially tricks the spliceosome into including exon 7 into doing the right thing.
We aren't fixing the DNA. We can't go into the vault and change the book. We are hacking the scribe. We are editing the editor.
And it saves lives. It turns what was a lethal disease into a manageable condition. It's unbelievable.
There's another distinction in Harper's regarding these errors that I want to clarify for everyone listening. The difference between a cis-acting defect and a trans-acting defect.
This is crucial genetic vocabulary. Think of beta thalassemia, the blood disorder. Often, the mutation is right at the edge of an intron in the beta globin gene. That is a cis-acting defect. Cis meaning on the same side. The defect is physically on the gene itself. It only breaks that one specific blueprint.
Okay, so if I have a factory, a cyst defect is like a coffee stain on one specific blueprint. Only the product from that single blueprint is going to be ruined.
That is a perfect analogy. Now, a transacting defect, trans means across. This would be if the splicing machine itself, say, one of the SNRNP proteins was broken.
So the robot that reads all the blueprints is broken.
And since that robot has to travel across to every single gene in the nucleus to do its job, a defect there breaks everything.
Yeah.
If your SNRNPs don't work, you can't splice any of your genes correctly.
That sounds incompatible with life.
It usually is. It's embryonic lethal. But interestingly, in the autoimmune disease, lupus SLE, the body starts attacking these transacting factors. Patients make antibodies against their own SNRPs. In fact, that's how Joan Stites discovered and isolated them in the first place, by using blood serum from lupus patients to fish out these molecular complexes.
It's a recurring theme in medicine, isn't it? Pathology illuminates physiology. We only learned how the machine worked because the body decided to attack it.
Exactly.
We spent a lot of time on mRNA, which is the superstar, but the genome is, 98% non-coding? Leninger calls it the dark matter. We used to call it junk DNA.
And it is definitely not junk. It's the operating system. We have rRNAs, ribosomal, and tRNAs, transfer, which we know are the workhorses of translation. But they don't just pop into existence ready to work either. They need massive amounts of processing.
Right. The ribosome is built from a giant 45S precursor RNA that gets chopped up into its constituent parts.
And it's heavily modified. You have to add methyl groups. You have to twist uridines into pseudouridines to stabilize the final structure. But the question is, how does the enzyme know which specific base to modify? The ribosome is huge. It's like finding one specific needle in a haystack made of needles.
It needs a guide.
It needs guide. Enter the sNRNAs, small nucleolar RNAs. They act like little seeing eye dogs. They contain a sequence that perfectly matches the target spot on the ribosomal RNA. They bind to it, and they drag the modifying enzyme to the exact right coordinate and say, put the methyl group right here.
So we have SNRPs for splicing and SNO-RNAs for guiding modifications. The cell's just full of these little RNA assistants, but then there are the assassins, the microRNAs, the mRNAs.
These are the silencers. They're tiny, about 22 nucleotides long. The cell makes them as a longer precursor. An enzyme called dicer chops them up, and then they get loaded into a weaponized complex called RASC, the RNA-induced silencing complex.
And what is the mission profile of RASC?
Search and destroy. The mRNA guides the complex to a messenger RNA that has a matching sequence. If it's a perfect match, slice. The RISC complex cuts the mRNA in half, and it's immediately degraded.
And what if it's just a partial match?
Then it just sits on the mRNA and physically blocks the ribosome from reading it. It puts a clamp on the translation process.
Why does the cell do this? Why would it go to the trouble of transcribing a gene just to send a hit squad to kill the message?
Fine-tuning. Speed. If you want to shut down a gene fast, you can't just stop transcription and wait for the existing mRNA to rot away naturally. That can take hours. If you release a flood of neRNAs, you can silence the expression of that gene in minutes. It's an emergency brake. It's also vital for development for sculpting tissues by making sure certain genes are strictly turned off in the wrong places at the wrong times.
This leads us to the final phase of the life cycle for an RNA, death. We touched on this with the poly A tail acting as a fuse, but let's look at the actual mechanism, the exosome.
Right, and we have to be careful not to confuse it with the exome, which is the coding part of the DNA. The exosome is the cellular shredder. It's a big barrel-shaped protein complex. Once that poly A tail is chewed down to a nub and the 5' cap is removed by a decapping enzyme, the RNA is fed into the exosome barrel.
And it's just ground down to its component nucleotides.
Exactly. Recycled, ready to be built into a new message.
Before we wrap up, there is one last piece of, weird biology in the outline that we have to talk about. RNA editing, not splicing, editing, literally changing the letters after they've been written.
This is the ultimate proof that the DNA blueprint is not destiny. In the human liver, there's a gene for apolipoprotein B. In the middle of the gene, the mRNA reads CAA, which codes for the amino acid glutamine. You get a massive protein called ApoB100. Its job is to transport cholesterol in the blood.
Okay, standard operating procedure.
But in the intestine, the cells express a special enzyme that finds that specific C in the mRNA and chemically deaminates it. It plucks off an amine group, and it turns the cytosine into a uracil. A C becomes a U.
So CAA becomes UAA.
And UAA in the genetic code is a stop codon.
Wow. It inserts a stop sign right in the middle of the sentence.
Precisely. So in the intestine, the ribosome chugs along and then just stops. Halfway through, You get a much shorter, truncated protein called ApoB48, and it has a completely different shape and a completely different function. Its job is to absorb lipids from your diet, not transport them in the blood.
So one gene in the DNA vault, two completely different proteins in two different organs, solely because the RNA copy was edited letter by letter on the fly.
It creates incredible tissue-specific diversity without needing to clutter up the genome with two separate giant genes. It's another layer of flexibility.
So let's bring this all back to the vault. We started with this idea that DNA is the static, permanent reference copy.
And I think we've seen that RNA is the active interpretation. It's transcribed by pol-totin, it's armored with a cap, it's timed with a tail, it's chopped up and rearranged by the spliceosome, and sometimes it's even rewritten letter by editing enzymes.
And then finally, it's shredded by the exosome. Its provisional life over.
The big takeaway for me is that biology values information flow and regulation over simple information preservation. DNA is the hard storage. RNA is the RAM. It's active, it's volatile, it's manipulatable, and that volatility is what allows life to adapt, to differentiate, and to regulate itself from moment to moment.
And a provocative thought to leave you with. We've talked about the spliceosome and the ribosome, arguably the two most important, most complex machines in the entire cell. And both of them are ribozymes. They are RNA machines.
It really makes you wonder, doesn't it? We think of DNA and proteins as the main characters of life, but maybe they're just the support staff. Maybe DNA is just a very, very safe box to store the RNA blueprints, and proteins are just the tools built by the RNA, for the RNA.
We might still be living in an RNA world. We just haven't realized it.
It's a humbling perspective.
If you really want to see the beauty of that lasso, the lariat structure of the intron getting cut out, open up Leninger, the chapter on RNA metabolism, it's a piece of pure chemical art.
Oh, absolutely worth a look. The diagrams are fantastic.
Thanks for diving deep with us.
Keep learning.